Jeanette, do you want to? I'm going to do the poo sample. That's probably more exciting. The poo sample, yeah. My name is Tagny Duff, and I'm a media artist. The project that I'm really interested in working on now here at BioArt Camp is to really rethink our relationship to different types of scientific instruments in the lab. And in particular, what I'm working with is the incubator. And the incubator is a fundamental instrument in the lab. It's used to grow agar plates, different types of bacterial culture, for example. And the aim of the incubator is also to remain sterile. You don't want any contamination in the incubator. And you want to be able to isolate different types of bacterial strains. And what I'm interested in is kind of polluting that relationship and bringing in the human, having a relationship with different types of bacteria, fungus, viruses, and trying to think about a relationship across scales, so across the human dimension to this microscopic world that we coexist with every day. But in scientific practices with different scientific instruments, we really try to isolate this relationship. And I want to see what new kinds of relationships we can have with different kinds of engagements with these microbes. So one way I'm doing that is to actually think about creating a new kind of incubator. It's kind of a crazy idea because I want to be able to create an incubator that biodegrades into the environment and actually contaminates the soil and melts into the earth and actually feeds microbes. So instead of using an incubator to remove any kind of contamination or microbial life, I want to make more life, more microbial life by feeding it. And so in a sense, I'm wanting to use different types of human body parts to do that. So for example, right now I've got my favorite instrument, the nail clipper, and I am going to be clipping my nails, collecting samples to create a kind of a buffet for the microbes. And also I've always got lots of dirt under my nails, and there's probably lots of bacteria growing in there too. So I want to mix that up and kind of make it a little more tasty for them. And I also have a bit of a cold right now, and so I'm hoping I can maybe incorporate some of my saliva and mix it in, and maybe introduce some new kind of cold viruses into the mix. Fungi are working with the microbial life around them, and no one is doing that research in the park, so I'll be able to isolate what types of microbes are actually in there.
So why is nobody doing that?
I don't know. I asked before coming here if I could work with someone, and they said no one in the park's interested in this. They're interested more in the wildlife, like the larger animals and the larger ecosystem. So this is going to be July.
So you can use this kind of samples for your durable labs?
Yeah, I'm gonna grow them, see what how the root system grows, and also what bacteria and microbes are around it. Mm-hmm. And then I'm gonna isolate that in the lab when I get back to Montreal. Yeah, I'll grow something. Grow a little mushroom maybe? A big mushroom? Like, that's a big mushroom, isn't it? I would like to grow one like that in the lab. Come on. Yeah, I need a better tool than this.
Mm-hmm.
I don't know.
I don't have something large enough to take this as a whole sample. Yeah. This one's cool, isn't it?
Yeah.
And there's some weird root systems here.
So giant.
Yeah, probably. Number 3. Let's go. So there's masses of spores on it, which I've got in a sample, and they're all yellow, and they're on the needles. So.
And you just found it by chance and looked it up as to what it was?
Well, he did. He saw it. I mean, like a true scientist, he goes back and he looks it up. Whereas I didn't know that that would be a mycelium, because usually I've only known it to be in the ground.
Oh, okay.
So they actually, like, they're a fungus that has a mycelium, and it incorporates the algal cells into it, and so the algae photosynthesize, and the photosynthetic products are used by the fungus as food. Actually.
Is it a lichen?
Yeah, that's a lichen.
It looks exactly like moss. So we've got the incubator going, and we're going to let it run for a couple of hours, and then we have to bring it inside. And the reason why we're bringing it inside is because we don't have the permit to allow us to keep incubating outside of the building. We have to move it inside tonight, and incubator. Then it's going to be put in petri dishes. They're going to be plated.
Sorry.
Talking to Angus the other day about how working in the lab changes your perspective on time.
Yeah.
And everything slows down. And you have to slow down.
Maybe 1 or 2 minutes more?
You think so? It's been in for 4 minutes.
Thank you.
Okay.
I was touching it.
Oh yeah, we'll get rid of that. So can you grab that? We'll go back into the lab. We're gonna have to put this on. The hardest part of this experiment right now is coming up with the right instruments to actually sterilize the medium that I've got here. So you can see what's happened. We tried parafilm over the container. Now this is saran wrap, and see what's happened. It leaked all out. It's just to be expected. So I've gone into the kitchen and I've got these Tupperware containers, and I'm gonna do it that way.
Oh yeah.
We're not gonna need this. We haven't used it, so.
But it's not. Well, is it on grams?
Yeah, it is. Wait. We got a reading. $200 water, and then 150
So that's
potato.
agar with just
Distilled water?
It's LB
no, it's
agar.
LB agar. So
Yeah.
it's agar mixed with nutrient solution from the lab already, and water. And this one has the homemade potato broth, which is the ancient way of making nutrient solution. So we're making a mess.
Stir stick, and probably stirred it a bit.
Okay.
But again, we're kind of improvising, so.
Stir sticks in over there in that Ziploc bag if you want to sterilize one with some.
Seriously, I'm a freak. Okay, here's number 1. It's very hot. We're in such different styles. So I know.
What did we do?
This is
Yeah, that's 6.
You got it?
Yeah.
Oops. Trying not to poke the agars. Yeah, there's nothing there yet. Because there's a lot growing, I can see already. So we want to make sure that they're sealed really well, especially if people are around. Out of the potato peels for our petri dishes that are going to have agar. They're agar plates. We need to make them to grow our samples. Often they're not really biodegradable, and there are some issues using corn-based materials because, of course, it does also use a lot of agricultural land space, and there's other ways, other materials that we could start thinking about using for biodegradable materials. I was experimenting.
Oh, I see.
It's a polymer like a starch and tapioca. There's tapioca beads and rice flour rice sticks. And it's just because I'm still thinking about what the substrate's going to be to build the shape for the incubator. But I do like this idea of working with maybe spruce and actually training or manipulating the growth.
Oh, I see.
The, you know, the wood, and then growing, the kind of the mycelium on top of it, and then so that way it's like different kinds of substrates.
Yeah, neat.
But that's what I'm going to have to test.
Yeah.
And it's like building something from scratch that hasn't been built before. So there'll be a lot of monitoring of the timing.
Gonna open it?
No. No. Because then for sure you're going to have microbes on your skin. One of my interests as an artist is also to not only kind of reappropriate different types of science tools and technology, but also the language of science. And so when I say contamination, I say it it's a bit tongue-in-cheek. I'm interested in playing with our fear around contamination, and so I like to emphasize these words that sometimes have negative connotations, but to playfully remove the kind of fear we have sometimes around words like contamination, pollution, sickness, illness, those kinds of words that have a stigma associated to them. And I do think with BioArt Camp, it's a perfect context for me to play with some of those fears around contamination and pollution, because I'm deliberately reappropriating those words and also the methods that are associated with those concepts.
Kiss the Jaeger plates?
Yeah. And take pictures of our mouth, this area here. We can stick the Jaeger plate to the right place, and you see the bacteria growing, forming your lips. It's very nice.
Well, yeah, okay. Well, that's kind of crazy. I thought that name was where we should have put the name up here. Then.
No, it grows like that.
So which one do we do it in? This one?
In the gelatin one.
Yeah. I'm wrong.
Follow me.
Already did?
Okay.
Walk this way.
Already did?
Okay, dear.
Thank you.
I can't get in there.
But you have no lips, that's why.
I know.
I get contamination.
Really? Okay, but when I'm working with HIV, I want to work with. Okay, so where are those stir sticks again? We want 3.5 to go in each separate container.
Yeah.
Oh, cool. I didn't think we were going to have this. Nice.
It's not weighing. So we're making a mess.
And what we're doing is we're going to compare and see which one grows the microbes faster. Yeah.
Just before you brought it in. And the autoclave lady puts it in this big pressure cooker, and tomorrow morning it's there.
Yes. Yeah.
And it's her job and her specialty,
And of course we have.
There are always a set of rules and parameters when you're working in a scientific context with scientific experiments. And there's often good reason for that. You know, there are times where scientific experiments can get out of hand and you walk into the unknown and certain things might happen that are detrimental. And then on the other hand, sometimes when you cross that line into the unexpected, wonderful things happen and new discoveries are made. And so, you know, what's happening here at Banff is that there are a certain set of rules and regulations that we have to follow. And on the one hand, it's important that we care for the environment and that we don't destroy the ecosystem. And so that's why rules have been put in place. Now, with the projects we're doing here, we kind of fall outside of the structure of a kind of a regular scientific experiment. And that can create some confusion, right? We're kind of testing the rules. And so projects like mine are not always so readily received. So I wanted to think about creating a wearable incubator that would be biodegradable and that would be made with different types of materials like found mycelium, different types of microbial life. And my original intention was to grow them and then create sculptures with different types of materials like potato starch and tapioca and kind of found kitchen materials and foods. And the craziest part is that I wanted to wear it and invite everyone to wear them too, and we would do actions in the forest, right?
It's like making a human popsicle.
Yeah. So I'm really thinking about what would it mean if we could completely reconceptualize our materials that we use in this kind of practice. So, you know, what would it mean to have a pipette made of mycelium that could then be, you know, thrown away after its use and then actually decomposed in a way that doesn't become part of the recycling method, but really does feed microbes and create another kind of ecology that's a kind of sustainable ecology. So you're all welcome to join me also in giving me feedback about your design.
Of your human popsicles?
Yeah, and also I'm interested in that discussion about thinking about the materials we're using while we're here in the camp, and also thinking about incubation and our you know, our concern for sterilization and containment, which a lot of us have to worry about, yeah, for creating these works. So this is the beginning of this particular project. Yeah, it's the conceptualization phase and just getting the samples, and then we'll go into the lab when I get back.
And design.
Another extended project where I'll be working with lentivirus, which is HIV, and using that to see how it interacts with mycelium, because there are some thoughts and hypotheses that there may be some kind of medicinal relationship where the mycelium can help block viral growth. People are coming to this bioart camp with many different projects, and I have yet to fully understand everyone's project. But I think what Jennifer's trying to do, in a way, is expose a public to how artists are asking questions about biotechnology, which is very different than how scientists would do that. Artists who are working with biotechnology and different types of biomedia are playing a role right now in creating a new media discourse, a new media art discourse. And what that means is that artists are bringing into the picture another way of thinking about media. It's no longer digital media, which has been predominantly the focus of a lot of discourse in new media art practice today. Now we're talking about different types of biotechnology that actually has been around for quite a while in the sciences, but artists are just getting a handle on it and able to incorporate it as media in their practice. And really, biotechnology is completely interrelated to digital media today. But we often forget about the wet lab practices and the wet technology that goes along with our digital representations and our digital constructions of our knowledge. So we need, I feel, we need to understand how this wet technology really is defining our everyday lives today, especially with the introduction of biosurveillance, different types of technologies that are surveying our every move around the globe, and different types of knowledge sets that are constructed and taken from biotechnology today. I don't know really what to expect when we go up to the camp, but I do know that there is a different quality here in the mountains, and that's going to impact what we do. I don't know, maybe I will have crazy dreams. Maybe that will become part of the project too. You can't discount that these environmental influences do have an impact on what you make and how you create knowledge. And in a way, I'm in a petri dish because the Banff National Park can be seen as a kind of an incubator of its own because it is so protected and also well looked after by the people who are in charge of the environmental health and safety issues.